Hello, good morning and good afternoon. Welcome to today's webinar, which is all about reproducibility, reliability, robustness, and establishing cornerstones for standardization of cell manufacturing. For those of you who don't know me, hello, I'm Nicola Ambler, editor of Facilitate Exchange, which is a digital content hub dedicated to advanced therapies and their commercialization. Today, we're going to be discussing which manufacturing parameters have significant effects on product performance, and then we'll look at potential strategies to mitigate variability. But before we begin, I'd like to say a huge thank you to the panel today for giving us your time. Sharing experience and knowledge is critical for the growth of our industry, so thank you very much. Also, a quick reminder to everyone logged in today that this is an interactive session, so your questions are very much encouraged. You can submit these using the question function that you see on your control panel in front of you. And then we'll address them all at the end of the presentation. I will hand straight over to our first speaker, which is Stephen Thompson of Sexton Biotechnologies. Thank you, Stephen.
Good afternoon, everyone. Thank you for joining this webinar today. So I'm going to talk a little bit about the need for robust process validation strategies in order to minimize the inherent biological variability that is associated with cell therapy manufacturing. So just to begin, a short overview of our focus at Sexton Biotechnologies. Sexton is a relatively new company that spun out of Incubator Accelerator Hub Cookridge Gentech in October last year, bringing with us a portfolio of technologies such as tools and media products designed for use in the cell and gene therapy space. Our focus is really to continue to develop aligning technologies which bring flexibility into manufacturing automation, ultimately with a view to increasing process efficiencies and reducing failures during the manufacture of ATMPs. Now, obviously, everybody is acutely aware of the complexity of cell and gene therapy manufacturing. The process of translating specific research into clinical effectiveness through development of a robust GMP process requires an enormous amount of consideration, and the decisions you must go through in order to develop such a strategy are extremely broad, not particularly standardized, and therefore require collaboration at multiple levels in order to validate processes. And the key points I'd like to touch on today is that whilst every ATMP manufacturer will be making these considerations and employing appropriate validations within their process developments for manufacturing of their own final product, it really is essential that therapy manufacturers demand the same level of process validation is carried out by any of their reagents or tools providers. With this in mind, the input material I'd like to focus on today is ancillary materials around cell culture. When undertaking a quality by design approach for process developments of an ATMP, it's essential to consider that the ability to produce an efficacious product in a consistent manner is very much dependent on the availability of quality materials. Therefore, it's important that due diligence is taken with each ancillary material to establish that they are available in sufficient supply at cGMP grade to support later stage clinical requirements, and also that the reagent manufacturer themselves has undertaken a robust process validation within their manufacturing to ensure quality and limit variability of their product. Because ultimately, ancillary materials will affect safety and, in some cases, efficacy of the final product. And so these product choices will lay the foundation for establishing a process that determines quality of the ATMP itself. Now, one of the challenges to try to qualify the safety and quality of ancillary materials is that they are not directly regulated. And so a lot is left to you as the manufacturer to decide what is the acceptable margin of inherent variability. And there's several factors which will affect this. So we have starting materials. So if it's a blood product such as a serum or a human platelet lysate, you need to consider where are the donors sourced from, what is the accreditation of the blood center, and what testing strategies are employed. When we consider the manufacturing process, we need to consider things like how large is the donor pool, are there any animal-derived products within the manufacturing, are there other process steps which may change the composition of the products. Also, is there a process such as irradiation to mitigate viral transmission? And how does that product compare to the non-irradiated product? What validations were carried out during the process development to document appropriate viral clearance but maintain adequate performance? And all of these factors will affect safety and quality of the answer material and also of the ATMP. And whilst guidances tell you how to think about that, they leave decision points fairly open-ended. And that can prove challenging. Unfortunately, cell therapy manufacturing is not like manufacturing of a pharmaceutical drug product, where combining materials A and B and C consistently produces product D. A significant proportion of materials required for ATMP manufacture are biological by nature, things such as donor starting materials and many ancillary materials. And unfortunately, biology is abidingly associated with inherent variability. And whilst there's no way to standardize these biological inputs, what we can do through robust process validations at all levels, from reagent manufacturer to the ATMP manufacturing process itself, is minimize levels of variability. And this begins with process design, whereby the commercial manufacturing process is defined based on knowledge gained through development and scale-up activities. We then move through process qualification, at which point the process design is evaluated to determine if the process is truly capable of reproducible commercial manufacturing. And finally, continued process verification. This acts as ongoing assurance, which is gained during routine production and ensures that the process remains in a state of control. And these are some of the ancillary materials which should have such process validation steps underpinning their development and manufacturing. FBS obviously has been used for many years, but is still inherently associated with lots of variability. There has been a shift towards chemically defined media in some cases, but it's not always been possible to replicate cell performance compared with biologically supplemented medias. Then we have the human-derived products. AB serum has become a gold standard for many immunotherapy-associated ATMPs, but there is certainly concern over the availability of a product which is derived from less than 1% of all blood donations. Furthermore, the varied lifestyle of donors, such as diet and level of exercise, often leads to lots of variability. On the human platelet lysate side, you have a choice between commercially available, ready-to-use platelet lysates or homebrew in-lab preparations. So there's a large choice of supplementations options for your process. And it's key to remember that these are product categories and not single products. FBS, AB serum, or HPL products from different commercial suppliers will likely be manufactured differently and hence will offer varying levels of quality and safety. It's therefore imperative to understand what process validations your vendor of choice has carried out in order to understand how it will impact your product. At Sexton, our solution is our range of human platelet lysate products. And again, we use the word range as we tend to think of this as a product category rather than a single reagent. We began with Stimulate, which is a pooled human platelet lysate, with large donor batches of approximately 100 units per batch, leading to limited variability. We then moved to incorporate a pathogen reduction technology, answering the requirement to ensure safety in the environment when you're not particularly sure which novel viruses may be evolving, and therefore testing strategies alone may not be sufficient. Certainly, regulatory burden increases over the past few years in Europe and Japan are moving to expect pathogen reduction in some of these human-derived ancillary materials. And now we're starting to evolve even further, so that we have cell-specific products such as Enliven, which is released against bone marrow MSC growth verification, and more recently our new T-Liven product, which is released against T-cell growth verification. So we're beginning to release based on relevant criteria as opposed to just random cell lines. And so when building out our validations during development of these products, we thought about with respect to three key attributes: performance, quality regulatory, which includes safety considerations, and supply needs. So can we ensure adequate supply to facilitate commercial manufacturing of cell therapies for our customers? From the regulatory safety side, as I mentioned, whilst ancillary materials are not directly regulated, guidances are pretty well defined. There is a need to ensure that your supplier is a robust supply of raw materials, that they are utilizing screened and appropriately tested donors, and that they limit any additives or certainly document what additives are used during manufacture. And that gets you to a good starting point. The sourcing of the raw material is a critical component of validation. Sexton obtained units from FDA AABB approved donation sites, ensuring that the platelet material was licensed as a transfusible unit, which ensures that you achieve a baseline safety profile. Other things to consider is how robust is a supplier's platelet donation network. Do they have multiple sites, which allows movement to specified sites should there be an issue such as a localized pathogen outbreak near one collection site? The quality considerations is the same sorts of aspects with typical quality system expectations. One key consideration is the ability to change tack with regards to ongoing regulatory developments. Certainly, European pharmacopoeia expectations of pathogen reduction and risk assessments of viruses really made everybody consider how they were going to do this with the platelet lysate. And if you're working with a supplier who's not able to collaborate and work with you to make the necessary modifications to satisfy your regulatory body, it may be that you run into supply chain issues during later stage clinical developments. And finally, performance. The material needs to be able to meet performance expectations for your cell type. As previous, it's key to consider the HPL, like all biological answering materials, on a product category, and different preparation methods could have a significant impact, not just on cell expansion rates, but also phenotype and efficacy of the final product. Another aspect in trying to improve safety and security of the human platelet lysate suppliers in the manufacturing processes themselves. Obviously, a GMP quality system is the foundation of this, but there's other things that you can do to improve the supply of the material and limit the risk from a supply chain perspective. At Sexton, we use outdated transfusible platelet units. Units are collected from a range of blood donation centres so that we have measures in place in case of any issues such as novel pathogen emergence or a force majeure. In addition, we have a large inventory of raw materials which allows us to quarantine units for a prolonged period before we introduce them into our manufacturing chain. Alongside that, ensuring full traceability of raw materials allows you to trace back any units that were donated after a given period of time where an unexpected infection arose, such as COVID-19. In our case, we are able to continue to manufacture for a prolonged period with units obtained before the outbreak. Other typical standard safety measures include limiting animal-derived materials and general good, clean manufacturing processes. So we mentioned the pathogen reduction processes that we've applied to two of our products. The current climate, where over the past few years we've had several novel pathogens emerge, with the original SARS virus back in 2002 and more recently Zika and COVID-19, it certainly adds thought into whether testing strategies alone of raw materials is sufficient. Certainly some European regulatory authorities and the Japanese regulatory authority require that human-derived materials be subject to a method of pathogen reduction. In essence, that seems a fairly simple thing to do. If you look into guidelines for finished product, guidelines around viral activation and removal are well discussed. ICH documents, WHO technical reports, FDA expectations. You can break down pathogen reduction processes into two general methods: inactivation or removal. For us, we began looking at inactivation as it's fairly well accepted for other answering materials such as FBS and sera. And so we began a full, robust development project to determine the best method for inactivating an accepted panel of viruses whilst trying to maintain product performance. We analysed several different methods, heat, chemical methods, a couple of different radiation methods, and we recognised as a complex biologic, it's very difficult to identify what you should be trying to protect from a product composition standpoint. Given we are already dealing with a product variable by nature, we didn't see any point in adding products such as stabilizers, which would undoubtedly infer their own variability. And so we eventually decided on using straight-up radiation. So both E-beam and gamma radiation are well-established methods to reduce infectivity of pathogens within a product. Whilst we initially evaluated gamma since it's widely used within the cell culture landscape, we found that using direct gamma radiation had a negative effect on the performance of our product with regards to its ability to support cell expansion. So we decided to move forward with a development project focused around E-beam. As you can see, E-beam has a very low penetration rate, and so we had to devise a proprietary method to ensure efficient viral reduction across entire batches of our product. After successfully developing this, typical validation requires you to spike the product with representative viruses. The guidelines from ICH risk assessment want you to consider a range of different virus types so that you cover both RNA and DNA, enveloped and non-enveloped, large and small viruses. Our findings demonstrated that we were able to successfully achieve an effective reduction of viruses in our products. Just to review these numbers, expectations in finished products states that log reduction in a range of 1 is not effective. Log reduction in the range of 2 to 4 is considered moderate to high effectiveness, and log reduction above 4 is a highly effective reduction process. So we were very happy with the results of the irradiation process, and by employing this development and validation approach, we were able to produce a product which had the same growth performance capability as the non-irradiated product. However, we did notice that the irradiation process inferred a positive effect when the product is used for expansion of CAR T cells, and there's more to come on that in a few slides. So, thinking about the ancillary material manufacturing process as a whole, ensuring robust validations through each step is the path towards minimizing inherent variability. During pre-development, there's a need to outline and understand baseline requirements. What are the critical quality attributes? These could be determined by the customer, a regulatory body, or internal interpretations of the need. Then, assign appropriate release criteria. Is it performance-based, i.e. cell expansion? Is it phenotype associated or maybe biochemical? Within these criteria, there's intravariability we must consider. How does cell expansion and phenotype vary between serum lots? Donor to consistency. How does cell expansion and phenotype vary between cell donors? Ultimately, here we're establishing a test method that produces repeatable and reliable results. Moving through to methodology, there's a requirement to begin to build test method specifications, identify process gaps, where to tighten controls of the process, how to reduce variability in the test method, begin to address raw material quality, cell line qualification, control serum qualification, and raw material specifications. Through these steps, we take all of the knowledge learned in pre-developments and developments to create a test method that will meet the necessary requirements in a validated manner. Now we have our validated process. Assigning in-process monitoring assures that we establish and maintain a repeatable, reliable, and robust process. And by working through these steps, we take something inherently variable by nature and, through robust process validation, develop ancillary materials with minimum variability both at the total protein level and also at the individual growth factor level, which is critically important for ensuring repeatable performance within an ATMP manufacturing process. Ultimately, all biological ancillary materials are a soup of growth factors, and we don't necessarily know which of them is responsible for a desired outcome, nor may we ever, as it's probably very likely multifactorial. But what we can do is ensure they are present in consistent amounts across varying lots of product. And this consistency within the composition of the ancillary material is notably translated into consistency of final product manufacturing. This recent publication with our colleagues at PCT demonstrates that donor-to-donor variability decreases by more than 50% from a cell expansion viewpoint when using Enliven as opposed to AB serum. Also, circling back to the surprise-associated effect of our E-beam irradiation process on performance of HPL and a CAR-T expansion, several groups have noted that the product drives a higher percentage of naive and central memory T cells compared with AB serum cultures. And this perceived improvements in phenotype translates to improved efficacy of CAR-T products in vivo, as demonstrated here by our colleagues at Baylor. Here, we see through this Kaplan-Meier survival curve representing liquid tumor model, the CAR-T cells expanded in different media supplements demonstrate significant differences in product efficacy and ultimately survival. And just as a final note, as previous, every biological product such as HPL or AB serum is a product category and not a single product. The development and validated manufacturing processes will have significant impact on product composition and ultimately an impact on the ATMP itself. This is demonstrated here, with Enliven expanded cells yielding a much higher percentage of cells with a naive and central memory phenotype compared with another commercially available HPL. So, in summary, there are many sources of variability within every single ancillary material manufacturing process that will affect quality, consistency, and performance of the final ATMP. Ensuring that your supplier has established a validated process akin to what you would have developed in-house for your own ATMP manufacturing process will ensure that all input materials you require demonstrate the lowest possible levels of variability. This is only achieved by ensuring reliable, repeatable, and robust processes are in place at all levels of the manufacturing process. So thank you very much for listening. I hope you found that useful. And with that, I'll pass back to Nicola.
That's great. Thank you, Stephen. Thanks for taking us through those studies and that data as well. And now we are going to pass over to Scott, our next speaker. Scott Jones is the VP of Scientific Affairs at BioBridge Global. Over to you, Scott.
Oh, sorry, I was on mute there. I think we're good to go. So thank you. I just wanted to start by thanking the organizers for allowing me to speak to you today. And what I'm going to talk about really is kind of build on what was previously talked about by Stephen, really a need for standardization of cellular therapy testing. Talk a little bit why that is, give a couple of examples, and then talk a little bit about lessons learned. We've been testing for several decades now for donor screening. We're now doing testing for cellular therapy, so sort of what we've learned through time, especially in donor screening and how it relates to cellular therapy. So I'm with BioBridge Global. Our mission is to save and enhance lives through the healing power of human cells and tissues. We have 4 subsidiaries listed here, and let me see, how do I get rid of that? And so we have several things over here. So we have a dedicated cGMP biomanufacturing space. We've been in the industry for several decades, over 45 years. We're a nonprofit organization, and we're headquartered in San Antonio, Texas. We're international. We serve customers on 4 continents, and we have a large testing facility. So we test over 9 million donations, which basically equates to over 65 million test reports per year. We do blood collections. We collect more than 130,000 blood donations every year, recover tissue donors, and also have collected or performed over 175 apheresis procedures. A little more specific about our capabilities. We really have what we call end-to-end capabilities. So obviously we work with, if you look on the left, we have source materials, we have our blood collections, we have different cell collections, we also have birth tissues, we do development, anything from cell therapy process development, cryopreservation, downstream, any part of the process. We do a lot with manufacturing, from preclinical all the way through to finish fill, quality management, and then testing. And that's really what I want to talk about today, is talk about our cellular therapy testing that's listed there in the top bullet points, and then also related to donor screening there, looking at plasma, blood, cord blood tissue, and organ donors, and really the safety testing we do for that. So so how I want to start off with is show you what we do for our donor screening. And as you can see here, there's categories. So we do the serological testing, which is basically antibody testing for these viruses. We do NAT testing, which is really looking for the viruses themselves. And we also do other testing, whether it's ABO-RH or looking for abnormal antibody screens. And so the key thing you can see here, and you'll see down here in the green that you see repeatedly, is the word standardized. Really, the take-home message here is that the work we do here is extremely standardized, from exactly what sample types we can do, what platforms we're able to use, what assays on those platforms we can perform, and then the general process, pretty much from start to finish, is all standardized. This is highly regulated, And obviously, most of it is dictated by either the FDA for the most part or other international regulatory agencies. So because of it, it enables us, because of the standardization, if we perform testing on samples and then those samples are sent to another lab running under these same standards, the results are very comparable between the laboratories. What I have next is looking at our cellular therapy applications. And so this is showing you the tests that we currently, the ones that we are currently running, the ones that we are bringing in and developing. As you can see, it's a very relatively large list. And what you can see is that we have multiple platforms, everything from flow cytometry, looking at surface markers, ELISA, PCR, and then other. They're also broken down into their different categories. The red ones here are characterization assays, the black ones are for safety, and then the blue over here is for potency. And really the key here is there are a lot of them, but there's really no standardization like we see for the donor screening that I showed you previously. In other words, it's really kind of the opposite extreme. There's really no standardization of platforms, assays, or processes. And the other thing, there's really no standard panel. In other words, when customers come to us, we're doing testing for maybe some of the flow cytometry, some of the ELISA and PCR. There's really not a standard panel, which is what we see in donor screening routinely. So why does standardization matter? Why do this? What I wanted to sort of give you is a couple examples of why we do what we do and why I'm here sort of pushing standardization today. One of those here is an ELISA assay. So we were interested, just like I showed you earlier, there are multiple assays that we need to do on ELISA for cell therapy testing. And we were looking at different platforms in order to do this. So what you have here are basically the results for looking for vascular endothelial growth factor, or VEGF. And we have two platforms. In the blue, you have the Luminex. It's a multiplex system, bead-based technology. And then you have MSD, which is an electrochemiluminescent system. These are both quantitative assays. And what you can see is we ran the same sample. We ran it multiple times. And technically, you should get roughly the same value. However, on several of the analytes like this, what you see is, in this case, about an 8-fold difference with MSD versus Luminex. To give you another example, this is sort of the same thing, but looking at P-selectin, same platforms. You can't really see the value, it's quite small, but there is a little bit of value there. But the point is, there's just a large variation just based on really the difference in what platforms you utilize. And obviously, this would make a very big difference in qualifying your product. And eventually, this is a product going into a patient. So this could have a very serious consequence or, yeah. So to give you another example, I just wanted to mention and talk a little bit about mesenchymal stromal or stem cells, what we're going to call MSCs. And I think Francesco is going to talk more after me in more detail about it. But bottom line is these are very cool cells. They're multipotent. They are stem cells, but they're non-hematopoietic. They're not immortalized cell lines. They are primary cells. But the key thing is the immunoregulatory, and that's why a lot of people are interested in them, as most people are aware. They have the ability specifically to sort of downregulate the immune response, which is a possibility for a lot of different therapies. And so there is sort of general criteria right now on MSCs. This is sort of defined by the ISCT working group. Very basic, but the bottom line is you have to show they're multipotent, that they can basically differentiate into fat, bone, and cartilage, that they're adherent, and then there's a series of surface markers that you look for. Some of them are the positive markers, and some of them are actually negative markers. And so what I want to show you here is an example of this. So this is basically taking human bone marrow derived MSCs from 3 donors. And what you can see here is we ran the positive markers, And we ran them basically for the same amount of cells grown, same conditions, except for the different media additive. So kind of like what Stephen mentioned, this is the fetal bovine serum. We have AB serum. We have 3 different commercially available platelet lysates, and then we sort of have another product we've been working on in-house, which is a platelet lysate type product. What you can see here is if you look at the markers, they're all positive. They're all expressing the positive markers. If you look at negative markers, same thing here. We just have the different negative markers on the left. But what you can see is all of the, the markers are not being expressed as what you expected. But let's say you look at function, so specifically looking at potency assays. So what we're trying to do here is look at the actual biological activity, right? The idea is we're expecting, hey, if they're looking the same on the outside, that they should have the same biological activity, but that's actually not the case. So what you're looking at here is a potency assay we developed in-house, which is looking at induction of regulatory T cells, so Tregs. The concept here is that immune modulation. This is a cell that that you want them sort of to induce in order to downregulate the immune response. And so what you see here is looking at the percentage of Tregs. So in other words, higher number means more Tregs. And these are the different media supplements down here below. And what you're comparing here is you're taking peripheral blood mononuclear cells, mixing them in with IL-2 to help induce, and also your MSCs. And what you're looking for is really a differential in the blue line compared to the brown and the green. And you can see that you get a little bit of level from fetal bovine serum, AB serum, lysate B and C. But you can see here in lysate A, it actually goes down, which means it's not inducing Tregs. But you look at your other 2 supplements over here, you actually see quite a large increase of induction. So even though they physically look the same, these cells, depending on how they were, what media supplement they're added, their biological function seems to be quite different. Why is that important with MSCs? Just briefly, cells change with different conditions for production and maintenance. So basically what I just showed, depending on what you add, and I think Stephen kind of alluded to it too, that depending on how you treat them, it will actually change their function. The other thing is this is a heterogeneous population. So you have multiple sort of subtypes. So what subtype are you actually looking at? What are you trying to characterize? You obviously have donor to donor variation. And really different sources of MSCs. So you could be getting them in the case like I just showed you, where it's bone marrow. You could also be getting them from fat tissue or other sources, and thus you would have to have some kind of standardization for how to look at them based on where they're from. So in sort of the last few slides, I just sort of want to give you kind of lessons learned that we have learned through all of our years of doing donor screening, but also what we've done currently with cellular therapy testing. And that really is that there needs to be standardization of test platforms, assays, and processes. Just like I showed with that example, if you don't have some kind of standardization, you're going to get very discordant results depending on what you're doing. And it could lead to big issues, especially, like I said, long term, the idea here is to do testing for something that's going to end up in a patient. So we really need to have it standardized, similar, in my opinion, to what we do with donor screening. So there needs to be standard tests for MSCs. What really defines an MSC? Obviously, the ISCT guidelines are good, but they aren't really enough. And like I said, Francisco will talk more about that. And this is critical. It's critical for really to make sure that these products that are going into patients are pure, safe, and potent. The other thing is really this concept of potency assays. As I say, potency, potency, potency. And the reason I'm saying it multiple times too is I don't think there's going to be one universal potency assay out there. You're going to have to do multiple assays. So I showed you one example of an assay for immunomodulation, but you're probably going to do that in conjunction with something like the IDO assay listed here, which is really looking at inflammatory signals or looking at differentiation, and also looking at cell health. And another thing to consider is, when do you do testing and qualification? Obviously, do it on the front end, and more than likely too, then doing it on the back end when you do release, but you're really going to do it at other parts in between. This is similar to what we do for our source plasma customers, where we do donor screening up front. They then do their further manufacture of their product. They send a sample back to us, and then we do testing prior to sort of the batch release of their product. Another big thing to think about are really these PCR assays. Most of them are listed, and most of the current testing laboratories are really using quantitative assays. And that's fine. The problem I have with it is you're not really making a call based on the quantity. Really what you're more interested in is there a virus there or not? It's really more of a yes or no safety issue, which is very similar to what we do in donor screening. And so with that, and the reason I kind of push it is, for example, if you look at Zika virus assays that are available, and someone did a study where they looked at all the assays that were available. Some of those were sort of your blood screening qualitative assays. Some of those were your diagnostic quantitative assays. And so they had 10 or 15 assays they looked at. What they noticed were, when you look at them all head to head, that the blood screening sort of qualitative assays were much more sensitive, much more reproducible, and really gave you a much better quality result. And so that's why I think in the industry, we really should look at qualitative versus those quantitative assays, and that's kind of what our group is doing. And then very briefly, the other thing is proper controls. This is a common thing we see when we're tech transferring assays from development companies, over to us to actually use for production, that you really got to make sure that you have good positive negative controls, possibly even an external control, to really control for all that variability like Stephen mentioned earlier. Appropriate validation, can't say enough about that. You really need to do a very thorough validation, and we kind of take the approach similar to we do for donor screening, where we're looking at reproducibility and inter and intra-assay variability and sensitivity. You got to do as much as you can do to make sure that it's really a validated system. And automation is another thing to consider. We currently, in the donor screening world, we do a lot of automation. The equipment is automated, and we're now also adding track systems to really automate the movement of tubes between. And it really adds a lot because it increases the quality, it puts more quality controls in there. Really helps with that reproducibility and really, kind of standardizes everything you do. And sort of my other comment is, don't be afraid of regulation. Cellular therapy is obviously a little bit younger industry than donor screening. And donor screening went through the same thing where people fought regulation and all those kind of things. But I think really the more you can work with regulatory agencies like the FDA, or look at, Pharmacopoeia and other European regulatory agencies, the better off you are in working with them and making sure that what's put in place does make sense and it's really controlling for what the issues are. So with that, I'd say thank you. I appreciate your time. If you have any follow-up after the meeting or whatever, you can reach me at this email address. And I just wanted to thank the people. So the MSC potency assay works. That was Anand and his group of Jenny and Kamali. And then the platform comparison was worked by Anthony. So with that, I will turn it back over.
Thanks, Scott. Thanks for delivering your insight and your experience from a donor screening perspective. And we will now move over to our final presenter of the day. But just before we do, a reminder for everyone listening that you can submit your questions using the question function on your control panel. So next up and final, last but not least, I should say, is Francesco Dazzi, Professor of Regenerative and Hematological Medicine at King's College, London. So over to you, Francesco.
There we are. I hope it is All right. Okay. So thank you very much for giving me the opportunity to share our experience with the use of mesenchymal stromal cells. I think what I would like to do with this is to add probably a further layer of complexity to the different factors that influence the reproducibility and the standardization of these cells. So, of course, Scott has already elaborated about this definition of the mesenchymal stem stromal cells. I think we're still quite behind at this loose definition of the ISCT, probably about 20 years ago almost, largely based on the ability of these cells to differentiate into the mesenchymal lineages. However, there are a lot of properties that we are leveraging out of these cells. They don't have a lot to do with these properties, but generally I would say that this is the preparation we use for clinical trials and clinical practice is a highly heterogeneous and not that yet well characterized therapeutic agent. And in particular, as I mentioned, the claim of this multitask therapeutic activity cannot, at least largely, justified by their stemness or the presence of it. I think that probably the most important property these cells exhibit is their ability to be immunosuppressive. When you toss these cells to T cells which have been stimulated with any type of stimuli, you can observe a very potent anti-proliferative and anti-inflammatory activity. Even at least in vitro, 1 cell out of 100 effector cells is sufficient to reduce the response of about 50%. So this MSC immunodation is probably instrumental at not only reduce this highly inflammatory activity in a lot of inflammation associated injury, but also as a result of this control to facilitate spontaneous tissue repair. So there are probably, I would say, probably 4 major type of MSC immunomodulation or actions. One is the direct local metabolic reprogramming. So these cells can produce enzymes that inhibit or sequester essential amino acids, or also the production of prostaglandin 2. They produce a set of anti-inflammatory cytokines, which also have a very potent immunomodulatory activity. There's been much interest in the potential of this extracellular mediated effect, although still is far less defined compared to let's say the standard cellular activity. And there is another very important aspect, which is the fact that they have a very interesting ability to engage with immunoregulatory networks of the host. I'm talking about myeloid suppressor cells or macrophages, but also, as Scott is mentioning, the possibility of directly or indirectly promote the expansion of regulatory T cells. So sounds like a very attractive package. And in fact, from what we know, at least in a small proportion of well-validated cases, MSC immunomodulation is fast acting and can be highly effective. It doesn't require any donor-recipient matching, which is very convenient. It can have an off-the-shelf reagent. This, of course, impacts on the fact that it is non-antigen specific, so modulating the immune system in a non-specific modality may raise or expose the patient to risks of generalized exposure to other pathogens. However, it seems that this immunomodulatory activity is site-specific. What I mean with this is that it is dependent on the exposure to inflammation. So if there is no inflammation, these cells become basically ineffective, or they are basically not primed to exploit their immunosuppressive properties. So I want to show how unfortunately, clinical practice is not following your rules, wrongly, I'm saying. This was a survey that we conducted on behalf of the European Bone Marrow Transplantation across different centers in Europe, including also Israel. What we found was quite, to a certain extent, disconcerting, because nobody actually followed any rule, or very, very few. This is a source, for example, of MSC. The majority is using allogenic, but some are also still using autologous as the only exclusive way of treating patients. Largely, this refers to practice for GVHD, but not necessarily the only one. Interesting that I think that there is, at least when we surveyed, there was still a majority of centers cycling the bone marrow. But I think everybody tends to move towards a different type, and it becoming, at least for logistical reasons, the umbilical cord tissue may be one of the most attractive, probably together with adipocytes, but because of adipose tissue-derived MSC is now, taken by Takeda, the other academic activity may concentrate on these other sources. Vast majority are using frozen cells, despite some investigators arguing that it may be better to have fresh cells, but there is no evidence, and it would be monumentally difficult to deliver fresh cells, at least across the world. So even the media used for manufacturing is quite different. At the moment of the survey, the majority were using pooled platelet lysates, but I think that everybody's moving towards, at least when it is possible, commercially available sources. Product specification is another a bit of a nightmare, and this is partly because there are, markers that remains a bit unclear why. Like, for example, HLA-DR, there's not much, point in doing it, but there is this ISCT criteria have been proposed to be part of the criteria, same for CD34, for example. But largely, probably the most important thing is that there are very few CD3, because that is where potential side effects could be generated. And these are the negative markers. As far as the positive markers, again, these are the typical three markers that I think everybody is using. There's not telling a lot, because these are exactly shared by any type of tissue fibroblast, but at least it has a good focus on the fact that the cells should not be hematopoietic or should not be endothelial, so vascular origin. Product specification, again, what is used as a release criteria? Karyotype at the moment was still used. I think it is less and less common, because we know that cells are saved. We know that the vast majority, I would say all of them, never been a proper report of chromosomal abnormalities. Majority are doing sterility, of course, mycoplasma, and also endotoxin. But this is not all, because endotoxin is something which is not yet entirely implemented. So I would say, fortunately, at least for those who are using the cells as an anti-inflammatory reagent, the tree lineage differentiation assay has been dropped, as well as the one investigating adipocytes or osteoblast. But the vast majority is actually not doing anything in this respect. Of course, bear in mind that if you want to do cartilage, you must do this type of release criteria, adopt this type of release criteria. Unfortunately, and as Scott was highlighting, it would be fundamental to have a potency assay. But at the moment, nobody is actually, or very few are using, because we still don't know exactly what we should investigate. So conclusion one, and just it's going to be, I would like to reverse a bit the perspective in my second part. But clearly, the manufacturing process and the release criteria remains highly heterogeneous amongst the different cohorts. But that is probably, such a heterogeneity probably reflects the still incomplete understanding of the scientific definition of the MSC product and why we want to use this. If it is, to build cartilage, you don't probably care about immunosuppressive activity, and vice versa. An important aspect is that actually patients are not considered into the equation, but they may still affect the clinical outcome. And I've come to this. So this is all what we have learned using MSC for graft-versus-host disease, a complication of allogeneic hematopoietic transplant, which complicates about 50% of the patients. It manifests itself with devastating activity on skin, liver, and gut, and the forms which are steroid resistance, which about 50%, are definitely lethal. So this is a brief summary. The only thing I want to say is that response rate, actually, despite the massive heterogeneity in the cell source, in the manufacturing process, and so on, remains about 50%. So we talk about between 30 and 60%. So despite using very different cell types. Another important aspect, because I think this should also be considered, this is a cohort patient that we investigated as a multi-center in the UK study, and probably the most important factor affecting response to MSC is the cell dose. The more cells we give, definitely the better it is. You can see that the response rate is almost 80% with 3 million per kilo, but if you use less than 1.5, is almost undetected. This could reflect, of course, a potency assay that we do not have. We may have done much better if we had selected some of these cells and make it more profitable, if you like, in terms of activity. Recently, we have identified a paradigm shift in the mechanism of action of MSC, which may inform us about the next steps to do. So what happens is that MSC are infused, and if the patients harbor activated cytotoxic T cells or NK cells, such as exactly in the case of GVHD, these cells undergo apoptosis. And these apoptotic MSC migrate to the tissues where they are phagocytosed by tissue-resident macrophages, which then are converted to become immunosuppressive by producing indolemin deoxygenase, which is, of course, the main effector of immunosuppressive activity. So the presence of cytotoxic cells in the patients seems to be fundamental to ignite the immunosuppressive activity. And in fact, what we did was to look at the ability of patients' peripheral blood to kill MSC before receiving MSC for treatment. And what we discovered is that patients who kill more are the patients who respond to the infusion. Patients who do not kill do not respond. And we have now a very robust biomarker whereby a cutoff of 11% killing predicts clinical response to MSC with a sensitivity and specificity of 90%. So it's very, very potent. And when we look at any other potential factors for response, the cytotoxicity is the only factor associated with response in a multivariate analysis. This also applies to Crohn's disease. We managed to have samples from Tygenix at the time of the trial for Crohn's, and exactly the same. So patients who killed were the ones who responded. Patients who did kill less did not respond to the infusion. So if the cells must undergo apoptosis or must be killed by the patients to produce a therapeutic activity, it is, I would say, almost obvious that we can elaborate a new potency assay whereby the cells which are killed more are the ones that should be given. So we can now, we do currently select our MSC batch according to the ability to be killed by a standard activated T cells. And these cultures must demonstrate MSC undergo apoptosis in at least 43% at least 43% of the cells undergo apoptosis. With this, there is still some variability, but at least we have a fairly homogeneous cell batch that is driven to elicit the mechanism we have investigated. So I think that the best practice and the best standardization should focus on a better understanding of the mechanism of action of the cells. It is fundamental to address the need for mechanistic biomarker to stratify patients, but also following the same concept, we have the opportunity to leverage the same concept to select the most potent batch, and of course potentially adapt the cell dose to the potency, because that would make a big difference. And we have a very solid readout to select which cells we should use as therapeutic activity. This, of course, the critical people in the laboratory who have contributed to this, to this summary. Thank you very much.
Thank you, Francesco. We've got a couple of questions in and are starting to run out of time, so I'll go through those for you now. And we'll go with a topical one first, and it asks, will COVID-19 have a significant impact on testing, and how have regulators reacted to past epidemics?
Is it to me, or where we can do? Yeah, okay, I'm on at the moment, so it's easy. So I think that, well, regulatory wise, I expect it will, because before we got the formal proof that the virus is not in the blood, we have to of course to consider this. But practically, I'm pretty sure there is, well, all the data at least so far have shown that the virus is not in the blood. It could be transient, of course, but I think that I don't think it should impact. And a PCR is probably more than sufficient, as we do for mycoplasma.
Scott or Stephen, do you have any thoughts to add to that?
Yeah,
so
go ahead, Stephen.
So I think at the moment, as I touched upon in my talk, obviously there's been several emerging pathogens over the past sort of 10 to 15 years, and obviously testing strategies can only basically analyze what they know. So I think obviously having a good pathogen reduction technology is obviously going to be paramount to making some of these products as safe as possible. Scott, did you have any more thoughts on that?
Yeah, the only thing I would add in is I agree with Francesco, meaning, right now we don't believe that you're having transmission through blood, specifically, through blood donations or transfusion transmitted disease. There are people, though, that are looking at that and evaluating it and putting it in models. And so I would say it sort of depends on how those results come out. But currently, yeah, the logic is that it really wouldn't be that kind of testing. It would just be the testing we're doing currently, whether it's nasopharyngeal swabs, looking for virus, or doing the antibody screens, which we do really for convalescent plasma. So really it's more for that, not necessarily for, true safety yet of products. That's it.
I'll ask the next question. I think it's more aligned to Scott, but if anyone else has anything to add, please jump in. The question asks, do you have any tips for embarking on an automation strategy for cell manufacturing?
Yeah, that's a good question. I can say what we're doing we're trying to look out there for technologies. I would say what I kind of envisioned in sort of basing it on what we see in donor screening is it would be really nice to have some kind of general platform that would be fully automated that, you know, could possibly do your PCR-based assays and your ELISA-based assays. I know in donor screening world, there's people that are working on that. The problem is they're quite expensive and that type of thing, but that's kind of what I would do. So to answer your question on the tips, I would basically keep looking, you know, the literature and talking to companies and looking for new technologies. That's been our experience, is it takes a while to sort of get caught up. But like I was trying to say earlier, the sooner you can do that, I think the better. But that's kind of what we do. And if you ever want to discuss it, just let me know. I don't know if my colleagues have any comments.
So I think from our side, Scott, certainly obviously at the moment there's multiple sort of automation platforms out there, and they tend to be sort of single units and very modular. I think what is really required moving forward is sort of flexible automation that allows you to integrate different components of a manufacturing system together.
Great. Thanks for your thoughts on that. And I will ask just one more. I know we've gone over slightly, but I think this wraps up kind of everything that we've been discussing today. The question is there currently a drive or a plan for different stakeholders, including associations like ISCT, to get together and develop a set of standards for the industry? I mean, is this even possible? Is it likely?
Well, to me, there is a risk of flattening the interest if we become too homogeneous. Although, of course, you know, harmonization is fundamental for little, you know, subsection of the manufacturing process. But, you know, having exactly the same cells, I don't think that anybody wants this.
So could you maybe break it down into categories rather than just try and do one blanket set?
Yeah, that is okay.
Stephen or Scott, do you have any thoughts on that, or do you know of anything that is currently happening?
I'll go ahead and speak then. Yeah, I know there are some organizations such as, because we're donor screening, we're affiliated with American Association of Blood Banks. So there's kind of a group there that sort of within there looks at, you know, cell therapies and is trying to looking at, you know, what we can do to sort of standardize. You know, I agree with Francesco to, you know, pretty much, but I think, yeah, there are groups that are starting to work on it. I don't know ISCT. I'm not sure if there's a specific group, but I think you're going to see that more and more. There's going to be publications and groups that are sort of trying to come up with that. But yeah, it's going to be interesting. And there obviously probably wouldn't be some kind of blanket one. I think there would probably be guidelines, you know, for certain cell types and very specific. And maybe even, you know, specific to different products. But to my knowledge, that's roughly all I know.
Thank you. Stephen, do you have any thoughts on this, or is it all being covered?
So I think that's largely covered it. I think there has been some discussion around standardization of HPL. There's been some good publications on that. But obviously everybody has different performance needs, and sometimes it's difficult to standardize the whole industry. I think what we need to get to is standardization of, you know, what are the required process validations for individual products that go into a manufacturing process that reduce that variability. I think that is the way that we can start to address some of the challenges of variability that we see.
Yeah, I concur. Brilliant. So we have run over a little bit, so I have to cut the questions off there. But I would like to just say once more a huge thank you to our panelists. We've covered some really important aspects of kind of managing variability today. And just for everyone listening, this webinar is also available on demand. So if there's anything that you need to refer back to or want to listen again, look out for the recording in your inbox later this afternoon, along with a certificate of attendance. We also got more webinars coming up in 2020. If you want to have a look at the schedule or keep up to date, just go to facilitate.co.uk/exchange. But I will leave it there. Thanks everybody for joining and listening, and good afternoon.